Hello everyone here. Today I'm going to talk about the papers that is named Bacteria-derived metabolite methylglyoxal, which modulates the longevity of C. elegans through TOR2 SGK1 deficit pathway. And here's the abstract that you could read about, and I'm going to talk about the introduction, some background first. So we know that gut microbe, it actually produces many different metabolites, including some short chains fatty acids that has both positive and negative effect on host physiology. And the Mg is actually high reacted decarbon metabolites that generated in metabolism of the human gut microbes. And also it has been found that some C. elegans will actually have generated in their intestine also. Mg also induce the formations of advanced glycations and products, AGE, through non-enzymatic reaction with some proteins, lipids, and DNA. And the main finding here, they find that the bacteria-derived Mg played a big role in modulations of the host longevity through TORC2 SGK deficit pathway. And the bacteria that produce less Mg, which is H and S, E. coli, and E. coli that overproduce Mg degrading enzymes, they extend the lifespan of the C. elegans through deficit. And also another point is TORS2 SGK1 were involved in these lifespan regulations. So involved in these mutants bacteria lifespan regulation of the C. elegans. And Mg also enhanced the phosphorylations of the SGK1 and accelerate cellular synthesis in human dermal fibroblasts, suggesting that there is a main role across the species here. And next, we're gonna look in the figure. So in figure 1, they first screen many gut microbes using C. elegans that growing off each of the 3,792 E. coli K12 single gene mutants. So K12 is actually set as a control there that we can see from figure 1a here. And the total standard normalized survival of the C. elegans in each pay would plotted against the p-value here. Figure 2b here. And they also have performed a secondary survival screen there. And finally, they isolated 17 pro-longevity E. coli mutants and 27 anti-longevity E. coli mutants that result in alterations in the survival rate, which is by around 20% in figure 1c here. And so also there is another point that overexpressions of the genes that shorten lifespan when muted has been reported to extend the lifespan in single gene or organism context. So that is why they also want to determine whether this also applies in interspecies context. So they did. They checked 24 different E. coli mutants overexpressing the gene associated with anti-longevity when mutated. And really interestingly, these bacteria generally enhance the survival of C. elegans. So this figure summarizes that this finding the genetic backgrounds of the gut microbes is actually a main regulator of the host longevity. So coming to figure 2, they already have screened up the 70 prone longevity mutations. So and the main gene here that they continue to work on is H and S gene, which is encoded H-and-S, is a transcriptional regulator that is highly conserved in the enterobacteria. And it actually regulates around 5% of the entire E. coli genome that is involved in metabolism, stress response, and also bacterial surface protein synthesis. So from figure 2a, we can see that the H and S E. coli mutants, when added to the C. elegans, actually extend the lifespan of C. elegans compared to the control. Control is using E. coli BW25113 parental strain, and we could see there is its extension of lifespan. So both H and S E. coli and BW25113 are of the E. coli K12 backgrounds. And one of them is set as control, which is BW. And from figure 2b and c is the health span tracking. So they first grow C. elegans on the H and S bacteria, and they check for the movement effect. So there is movement assay here and heat stress resistant assay. And it seems that there is no effect on the movement assay. So it's not affecting the developments and also dietary restrictions here. And they find that the C. elegans grows even faster, and the reproduction is not affected here from figure 2d. And this data indicates that H and S not only extend the lifespan but also improve health span of C. elegans here. So next, they're gonna have some insights on the mechanisms. They want to determine whether H and S extend the lifespan through well-known longevity pathway, which is step 16 here. There is many mutants they use. DEP16 mutant TOR, REG-1, and DR mutants, dietary restriction mutant E2, SIR 2.1, immunity mutants like PMK, and also stress response mutants as scheme 1, right? And HSF-1, JNK, GK7s. And interestingly, that they find the lifespan extension by H and S was completely lost only in the deficit mutants, suggesting that the H and S activates C. elegans deficit to extend the lifespan. But which pathway is actually upstream of the deficit? That is what they want to find in the next part. So we also know that C. elegans will enter downward depots which is dependent on deficit. So to confirm that H and S E. coli activate deficit downward formations was assessed in the IS compromised DEF2 mutants. So the DEF2 mutants is growing on H and S that from 78% downwards at a temperature of 20 degrees Celsius compared to 24% downwards when grown on the control E. coli. Moreover, in figure 2G that we can see here, they find that there is an enhanced heat resistance by H and S E. coli, which is deficit independent. So from this message, we can summarize that H and S E. coli activate C. elegans deficit to enhance the longevity, downward formations, and stress resistance. So next, we're gonna further investigate that whether there is other pathway there. So they have 5 E. coli mutants. Initially, that they screen H and S, HYFR, and UIDC, LON, and SAPD. These mutants has been previously reported to produce a high level of the extracellular polysaccharides Ca, and this stuff extend the lifespan of the C. elegans through activations of the mitochondrial longevity pathway, which is UPR MT that we can spell in short term. So unlike other mutants, the contribution of the Ca to the lifespan extension by H and S E. coli is partial. And this means that H and S extend the lifespan through both Ca dependent and could be also Ca independent mechanism. So to first confirm that H and S E. coli mutants can promote longevity of C. elegans in independent manner, they knock out the CP, SB, and GALE, and UGD genes, which is from figure 3ABC that we can see that they are all encoded for the biosynthesis genes of the Ca precursor. So they find that none of these mutations led to complete suppression of the lifespan extension by H and S E. coli mutants. which means that this is actually something that independent of CA. I mean, for the lifespan extension of the HNS. But they further introduce mutations in the CA biosynthesis enzyme in the HNS mutant background, including mutations of the WCA DEF, which encode the important CA repeating unit polymers. And they find that these mutants actually is stretching the CA-dependent lifespan extensions of the NHS C. elegans. So, in contrast to the lack of lifespan extensions by CA in C. elegans mitochondrial longevity mutants, they find that HNS mutants extend the lifespan of all mitochondrial longevity mutants. So, from figure 3A to figure 3D, this part message that summarizes that HNS E. coli mutants is able to extend the lifespan of C. elegans through E. coli CA, C. elegans UPR mitochondrial independent mechanism.